Good afternoon. We are here in Philadelphia at the ACR conference with Professor Francesco Del Galdo. And so, Francesco, could you provide us some highlights about the biological diagnosis of scleroderma in patients with VEDOSS?
Yes, we just finished the presentation here. VEDOSS is a project started a few years ago, and we published in Lancet Rheumatology only earlier this year, and it showed the proportion of patients that are at risk of scleroderma that they progress to disease. And it's actually a very high proportion if they have scleroderma antibodies and capillaroscopy changes is as high as 75% within 5 years. So this triggered the idea that perhaps the diagnosis is limited by our clinical detection threshold, and scleroderma is already happening. We took biopsies from these patients and we gave them to a pathologist without telling them where they're coming from, and they confirmed they were scleroderma. So even if we could not detect any screen thickening, muscle trichome, CD45 staining, and H&E looked like scleroderma. And we did more. We cultured fibroblasts out of them, and also fibroblasts maintain the same profibrotic activation that we know is typical of scleroderma. Also in their blood tests, we checked for interferon score, which have two other posters here showing how important is type 1 interferon to predict severe outcome. And this was very interesting because patients with VEDOSS fit exactly in between healthy controls and patients with scleroderma. So from the type 1 interferon point of view, they are brewing the disease. So this led us to think that perhaps if we look at biosamples, we can diagnose scleroderma biologically before they fill criteria, and this will inform prevention trials.
So very important results for the field of scleroderma. Thank you.